Hello, everyone. My name is Claire Stevenson, and today I will be expanding on my summary of the biochemical pathways that I made for Dr. Callaway's nutritional biochemistry class for the midterm. And today for the final, I'll be discussing a little bit more about these pathways, just in terms of where different sugars can be shuttled in, as well as where intermediates are used for fatty acid synthesis and where amino acid metabolism can also fit into these pathways. I'll then talk a little bit about DNA and RNA synthesis as well as the urea cycle. So let's get into it. First things first, when we're thinking about glycolysis, glycolysis is incredibly important for bringing glucose into the cell as well as converting glucose, which is a 6-carbon molecule, into pyruvate, which is a 3-carbon molecule. This pathway is pretty universal in its intermediates and enzymes between glycolysis and gluconeogenesis, where glucose is made from a non-carbohydrate source. Um, but one thing that I want to touch on that is new is the fact that galactose, fructose, and mannose can also be shuttled into this pathway. Galactose can be converted into glucose 6-phosphate via the lower pathway, and fructose and mannose can be converted into fructose 6-phosphate to then also be used as potential sources for these intermediates as well. Or these intermediates can be broken down into these other sugars if those sugars are necessary for a different purpose. In terms of the anabolic and catabolic metabolism of amino acids, this is mostly going to fit into the citric acid cycle, where those will be where the amino acids will be broken down or converted to acetyl-CoA, pyruvate, alpha-ketoglutarate, succinyl-CoA, fumarate, and oxaloacetate. So the different ways that these can be shuttled in is mostly going to be at the top here into pyruvate and acetyl-CoA. Alanine will be converted into pyruvate. Threonine will be converted into glycine, which can be converted into serosine, which can then be converted into pyruvate. And cysteine can also be converted into pyruvate. Threonine can also be converted into acetoacetate, which is an intermediate before acetyl-CoA. Lysine and tryptophan can also be converted into this intermediate before becoming acetyl-CoA. Leucine, tyrosine, phenylalanine, and isoleucine can also be converted into acetoacetate. Tryptophan can also be converted directly into pyruvate instead of going into the pathway as acetyl-CoA. Then when considering the amino acids that can be converted into alpha-ketoglutarate, we have glutamine and glutamate and glutamine, which use the same metabolic pathway, as well as histine, arginine, and proline. Arginine and proline are also going to use the same transamination to be converted into alpha-ketoglutarate. Then threonine, valinine, and methionine can also be converted into succinyl-CoA. And then phenylalanine can be converted into tyrosine, which is then converted into fumarate. And asparagine and aspartate can then be converted into oxaloacetate. So this is just one of the many ways to show the complexity of biochemistry and how it's very fluid and ever-changing, just depending on what the organism needs. So let's say the organism really needed energy and it didn't have a lot of intermediates in order to create this energy, then these amino acids can be broken down through catastrophe, breaking down, to then be converted into these intermediates into this very important pathway for producing energy. Or if we're trying to create any of these amino acids, we can use these intermediates as a source to create the amino acids. So it's just, again, like I said, showing that fluidity of biochemistry and really overall just another reason why the mitochondria is the powerhouse of the cell, because all of this is taking place in the mitochondria, as this orange line points out. And so all these amino acids can be shuttled into the mitochondria if necessary, or they might already be there. And then they can be used to create energy during the citric acid cycle, which is used to create reducing equivalents in the form of NADH, FADH2, that is then shuttled down to the oxidative phosphorylation pathway. In this pathway, I'll just briefly mention that Oxygen's electronegativity helps to pull electrons through these different complexes. As the electrons are being passed through the complexes, then hydrogen atoms can be pumped into this intermembrane space, which then makes it energetically favorable for hydrogen atoms to be pumped back into the mitochondrial matrix using the ATP synthase to then produce a massive amount of ATP, maximum up to 38 ATP, in fact. Down here in glycogen, glycogenesis and glycogenolysis, we just have the buildup of glycogen from glucose, anabolic process, or the catabolic process of breaking down glycogen into glucose, depending on the needs of the organism at that time, if we need to store the glucose or if it needs to be used. But nothing really new is being added here. In terms of fatty acid synthesis, I didn't really talk about this last time, but glyceraldehyde 3-phosphate, G3P, is an important fatty acid synthesis intermediate. And I did briefly touch on how NADPH is used for fatty acid synthesis as well. Last time I did talk about how ribulose 5-phosphate is used for nucleotide synthesis. So that's what we're going to talk a little bit more about now. So ribose 5-phosphate is converted into PRPP, which is the active form, with the enzyme phosphate diphosphokinase, and PRPP stands for phosphoribosol pyrophosphate. And from here, the PRPP can then be used to make the nitrogenous base of DNA, either through de novo synthesis to make a purine or the salvage pathway to make a pyrimidine. So, just to kind of think about DNA in a very general sense, you have your sugar ribose backbone, the phosphate groups that connect this backbone, and then your nitrogenous bases over here. And so, if you're thinking about rungs on a ladder, if you're thinking about DNA as a ladder, excuse me, then these rungs on the ladder are the nitrogenous bases, which are being synthesized from the ribose 5-phosphate, converted into PRPP. And then the sides of the ladder, I guess, is the base of the DNA, which is the negatively charged phosphate groups connecting these ribose backbones. And just to kind of specify here, the only difference, well, major difference between DNA and RNA is the placement of the hydroxyl group, either on the 3' carbon or the 2' carbon. If it's on the 2' carbon, like it is in RNA, then it makes it more reactive. But anyways, to get back to the nitrogenous base synthesis using ribose 5-phosphate, we can either make purines, which are a 2-ring nitrogenous base, and that is adenine and guanine, or we can make pyrimidines, which is a single nitrogenous ring, and those are cysteine and thymine. Cysteine and guanine are connected by 3 hydrogen bonds, versus adenine and thymine are connected by only 2 hydrogen bonds. So that's why like the Tata box is where DNA replication usually starts, because it's easier to break apart the DNA where there's a lot of A's and T's, because there's only 2 hydrogen bonds between those 2 nitrogenous bases, unlike the 3 hydrogen bonds between guanine and cysteine. So in order to do this, we're going to take R5P, which is ribose 5-phosphate, convert it into PRPP, and then convert it all the way down here into IMP, which is the complete purine ring. And like I said, the purine is the 2 nitrogenous bases, and that's why there's 2 nitrogen rings here. And then from here, we can also make the single nitrogenous rings of the pyrimidines using the salvage process or the catabolic process of purine. So it all just kind of depends on which way you're looking at it. It could either be, like I said, the catabolic breakdown of this purine nitrogenous base, or it's the anabolic buildup of the pyrimidine nitrogenous base. And then from here, IMP serves as a critical component to be able to create these rings that create the nitrogenous bases to be able to connect the DNA backbones. And then what's important to note is when you consider your carbons, you have carbon 1, 2, 3, 4, 5. And so you have this 5' to 3' orientation. And then one side is going to be going one direction and the other side is going to be going the other direction. And this is really important to note when you think about DNA replication, because one strand is going to be your leading strand and it's going to have continuous replication because the polymerase reads the same way as the actual base is. But the other side that's going in the opposite direction is going to have these Okazaki fragments where it will only be able to synthesize a little bit before going back to the beginning again because it's maintaining that same orientation of reading from the same direction. Um, but during this DNA and RNA synthesis, uh, that's where you create these bases in order to be able to have that replication. And so then if you're going to, break down the DNA or RNA molecules, you have to first be able to rip apart the latter. I know double-stranded RNA is not very common, but it is a thing. But for the most part, when you're talking about DNA synthesis, then you're mostly going to be talking about double-stranded DNA, which is two-sided and has to be split apart in order to break down the actual DNA parts. Anyways, I digress. Let's get into the urea cycle. So in the urea cycle, you have four major steps. So you first have transamination, which is basically just rearranging the molecule. And then when you take alpha amino nitrogen and convert it into ammonia using a glutamate transdeamination and GDH, then that is the oxidative deamination process. And then from there, um, we're going to have an oxidized amino acid that then helps to release an NH3 in the liver. And then the NH3 and CO2 is what's entering the actual urea cycle here. So in the urea cycle, you're first going to start in the mitochondria of the liver and then end in the cytoplasm of the liver. And you're going to end with converting all the way to urea, which can then go to the kidneys. So the purpose of this is to then prevent ammonia intoxication. It's a way for us for our bodies to biochemically get rid of excess nitrogen, either in the form of ammonia, urea, or the uric acid. So from this carbon dioxide and NH3 molecule, the enzyme carbol phosphate synthase 1, which is the rate limiting enzyme, is then going to convert the molecule into carbonyl phosphate. Carbonyl phosphate is going to combine with orthenine, using orthenine transcarboxylase to make citrulline. And then citrulline will make arginosuccinate, which is what provides the second nitrogen for urea, before arginosuccinase converts it into fumarate and arginine. Arginine then is converted by arginase into urea with the help of water. And so then there are 6 different amino acids that are used in this full cycle. But ultimately, we're trying to get down to that simple urea that can be excreted out in the urine, and that's why it's going to the kidneys. And this, like I said, helps to prevent ammonia intoxication. And that's why if you're feeding too much nitrogen to an animal, you should be able to smell it in their urine. And this is why, because it's being shuttled to the kidneys. So I hope I was able to answer all of the big burning questions that you have, Dr. Callaway, for our final. But otherwise, I just wanted to say thank you for a wonderful semester. And I really did enjoy learning more about biochemistry. Um, as you can tell by my in-depth document here, I did really actually enjoy learning more about the biochemistry. I'm a bit of a biochem nerd, but anyways, I hope this answers all your questions, and I hope you have a good rest of your day. Thank you.